Hello, hello. So you have met Jose before in the field. He looked at our plot to field sites and collected a few bugs. So we wondered what he's up to with those bugs. So here we are at my Evenden's lab at University of Alberta, so Biological Sciences Department, and checking on him, what's going on. So here's Jose.
Hello. Yes. So last time I was in a tall Peaola plot, having the time of my life. Unfortunately, the field season has come to an end, but the work for me is just beginning. So we're here in the lab and we're taking a look at sticky cards. So these little guys that we saw from our plot to field sites are what I'm going to be looking at now. So we're actually at the lab, we're going to be looking at these, and we're actually going to be able to see the number of insects that we're capturing. So we're at the stage where we are trying to identify and count what we actually found throughout the summer.
So far you've been able to find a few of our predators and parasitoids and the major insect, which is the diamond black moth.
Yes, yeah. So we have been finding very few numbers of diamondback moths, a lot of parasitoids in these plots. So obviously once we actually do the stats behind everything, we'll see if there are already differences. But we are finding insects that we are interested in, which is the major point of it, which is good. So the way we go through these sticky cards is pretty simple. We want to put them underneath the dissecting microscope. And there's grids in the sticky cards, so we just go down a grid and go to the next grid and keep on going down the grid. Essentially just trying to look at all the insects that we have in there. Once we come up to an insect that we like, a parasitoid, we'll have a clicker and we'll just click it. And we'll keep count of how many parasitoids, hymenopteran parasitoids we find, how many diamondback moths we find, how many flea beetles. We're really looking at a lot of different insects, so we want to keep track of all those different insects.
And this is one part of what you established as an experiment in our trials. You also have collected some vials, and these are the live samples, I believe?
Yeah, so these are the sweep samples. So in each of those plots, I conducted several sweep samples throughout the season. They're in this little bag here, and we put this bag in the freezer to kind of preserve the insects as best as we can. Once we have this, then we can take a nice little tray that we have here. We dump the contents of our sweep net sample into the tray. We put the tray underneath the microscope. We take our trusty paintbrush, which is really good for separating out insects from the plant. I know it's not exactly a very scientific tool, but it gets the job done. So we'll dump everything in there, we'll sort everything out, and sort of like what we do with the sticky cards, we want to keep track of what insects we find. So we have these little vials with ethanol that we put all our insects in. So we separate them into orders. So all the beetles get put into a vial, all the flies get put into a vial, all our moths and butterflies into one, bees, wasps, and sawflies all get put into a separate vial. And the goal of this is to have each of these insects separated by order.
Yeah.
So then we can come later and identify those insects to families to see what specific families of insects are we actually seeing in these plots. And that'll help us get a better understanding for whether they're parasitoids, pollinators, natural enemies, or just general insect pests. So here I have a nice little vial filled with a bunch of larvae of Lepidopterans, so caterpillars. And so these will be looking to identify as well as part of our functional groups.
Very interesting. So, any first thoughts, anything that you've been observing so far?
Yeah, so far we've been seeing that there hasn't been many changes in diamondback moth preference. So we're seeing kind of no preference, so same in monocultures versus intercrops. But obviously we're still having to go through all the data, so it's very possible that there will be some changes. Same thing with parasitoids. We are seeing preference for some plots versus the others. But again, it's hard to say without fully getting all the data and really running the stats. Right now, this is just very preliminary analysis of stuff.
And for after hours and hours of this microscopy work and identifying all of them, you're going to go sit in front of a computer and punch in your data.
Yes.
And then put a statistical program to analyze what actual differences you saw.
Yes, exactly. You go from staring into this to staring into something else. Unfortunately, that's how it goes sometimes. But yeah, so this is just, again, one part, like Jagroop mentioned, of the whole process, right? We collect the samples in the field, we identify and analyze the samples in the lab, and then we run all the statistical stuff on the computer And the stats is what's actually really going to tell us whether we're seeing any meaningful differences between those groups. So obviously we have our plot to field sites, but in the lab we can also try our best to replicate plot to field and intercropping to the best of our abilities. So we have some growth rooms set up upstairs that we can grow both canola and peas together in an intercrop and introduce insects to these containers to measure different aspects of their behavior. So right now we're running a herbivory experiment where we're setting first instar diamondback moth larvae onto canola plants and pea plants to measure how much damage and how much feeding they're actually doing in these intercrops versus the monocultures. Additionally, we're waiting for them to pupate. So once they pupate, we're able to remove the pupate from these plants, weigh the pupate to see if the feeding or lack thereof impacts the pupal weight, which can ultimately impact adult characteristics. So once we get them once we pull the pupate, we put them in little containers, we weigh them, we wait for them to emerge, and once they emerge, we can freeze and kill the adults, and then also get characteristics from those adults. So the weight, the sex, the length, and the wing area are all very important characteristics of these adults. So removing the pupae from the plant is really important because we're able to then essentially put a little collar on these larvae and identify which larvae or which pupae is the adult coming from. So then we can tie the pupal weights to the adult weights and adult characteristics. So once the larvae have done all their feeding and have pupated, we remove all the leaves from the plants and scan them. So we're able to quantify how much damage they're actually doing to these plants. In addition, it's also important that we look at plant characteristics. So at the beginning of each experiment, at the end of each experiment, and throughout, we're keeping constant tabs on what growth stage the plants are in, and the height of those plants, and how many leaves are in each of these plants, because that's also important information from an intercropping perspective. How well are these plants doing when they're grown with peas? So it's important that we get all aspects, the plant side of things and the insect side of things as well.